Have you introduced Kristina? So
No, we just started.
We're just starting. Okay,
yeah.
Thanks.
Would you like I do? I go ahead?
Yes, please.
Okay. So welcome everyone at the Imaging World. It was a while that was not attending. I'm grateful to be here today. Today's speaker is Kristina Ganzinger, who received her PhD in physical chemistry from the University of Cambridge in 2015, under the guidance of Sir Professor David Klennerman. After a postdoc at the Max Planck Institute for Biochemistry with Professor Petrashvili, one of our speakers in the past, she began her current position as tenure track group leader at the NWO Physics Institute in the Netherlands in 2018. So the central questions driving her research are actually things that I feel very close to, is how do cells transmit, process, and respond to information, both precisely and unambiguously, and how do they shape their membranes in response? If we understand the molecular mechanisms of cell signaling, can we design and tune artificial signaling networks? So her lab's approach is to address these questions by in vitro reconstitution in model membrane systems, what she refers to as minimum cells, and pairing these studies with live cell experiments. So they've done quite a lot of custom microscopy, both in terms of analysis and engineering, particularly new single molecule microscopy methods. So anyway, without further ado, Kristina, the stream is yours, and I'm looking after your talk and the question afterwards.
Yes, thank you very much for this kind introduction. In fact, this talk is really very much going to be on single molecule method development and also a bit on the technical side. I'm just sharing my slides. Here they are. So I will talk about how we use DNA-PAINT not to actually do super resolution imaging, but to do single molecule tracking in cells, or also in reconstituted membrane systems, as was just mentioned. That's something that we are very much interested in and working with as well. Yeah, and I also really want to thank the organizers for this organizing the seminar series, because I think it's actually a really great idea to have an international series like this that is centered on microscopy, which of course we all love. So Alessandro has already kindly told you a bit about what we do and what we are interested in, but I still want to introduce myself and my group a bit to you. So we are really interested in immune cell communication and how these cells, when they interact with one another, pass signals from one cell to the other, and how they then process these signals to respond to them, because as we all know, in the immune system, this is often a matter of life and death, not even just for the cells, but also for the organism. And we kind of have two approaches. One I call a hybrid cell biophysics approach, because we still use reconstituted systems, supported lipid bilayers, to interact with immune cells, but then we actually follow what these immune cells do in response to these well-defined inputs, still doing like in-cell imaging. And these are just some examples from my research with David Klennerman, actually in the past, on immune cell signaling, where you see CEP70 binding to T-cell receptors as they are activated in kinase-enriched contacts. And on the other side, we also follow a full bottom-up reconstitution approach, where we really try to build up these signaling pathways from their molecular components in membranes, both vesicles and supported lipid bilayers, to really get a better understanding of the biophysical principles behind this information processing. And of course, I wouldn't be giving this talk in this series if it wasn't for the fact that our working horse method really is fluorescence microscopy, and often single molecule fluorescence microscopy. In the first part of my talk, I will talk about the DNA-PAINT single particle tracking. And in the second part of the talk, I will talk about how we want to use this to study interleukin signaling in model membranes. So this is a quotation that I found in a PhD thesis from a PhD committee that I was recently on, that I really liked. It says, the length of the particle's trajectory in single particle tracking is of utmost importance, as it provides the necessary data to detect any variation in the particle's motion. And so also anything that happens to that particle, as it, in this case, for example, you can see the binding of an adapter protein. And as you can maybe appreciate from this movie already, this is actually a really long trajectory. Like at least those among you who do single particle tracking know that often we can maybe only get trajectories of 10 to 50 frames or so. In this case, it's over 500. And if I would tell you that these two trajectories overlap, the white and the purple one, I think I wouldn't find it very hard to convince you, just simply because this overlap is so long that it is really unlikely that both of these molecules would diffuse in this exact very same pattern just by chance without there being any interaction between them. But to get there actually was quite a long story. And we really, because these really long trajectories are so important to us if we study single molecule interactions, we were really unhappy with the state-of-the-art labeling methods for single particle tracking. Because on the one side of the spectrum, you can label your molecules with organic dyes or fluorescent proteins. Because of course, what you actually follow with the single particle tracking is not the molecule of interest, but actually the label attached to it. And these organic dyes or fluorescent proteins, they are all limited by photobleaching in the sense that once they are bleached, they are out forever. And they give you a very limited observation time window of like a second or maximum maybe 10, 20 seconds in the very best case scenario. And on the other side of the spectrum, you can also label your receptors or whatever you are interested in using inorganic structures such as quantum dots. They don't bleach, but then they blink. So you still have the problem of truncated trajectories because of that. But they are at the moment the only way to get really long, or they were the only way to get very long trajectories on the time scale of minutes. The other disadvantage that they have is that they are actually very big, and it's really difficult to just attach a single molecule to them because they are orders of magnitudes larger than a single molecule. So our new DNA paint-based method sits in between in terms of size. We actually have a DNA label that then gets labeled by single-stranded DNA molecules that are attached to a fluorophore. And the nice thing is that it really also gives us minutes of observation times for individual trajectories, as you will see later. So the concept is actually reasonably simple. What we do is we mark the molecule covalently with a very long DNA strand that has multiple binding sites for the same short DNA stranded molecule called imager. This is very similar to using DNA paint also for super resolution microscopy, with the exception that we don't have a single binding sites where these imagers exchange, but we have multiple on the same molecule. And in the end, this allows us to get these really long trajectories because we constantly have at least one imager bound. And this work was actually led by my PhD student, Chris Niederauer, in collaboration with Petra Schwille and Ralf Jungmann at the Max Planck Institute for Biochemistry in Germany. So as I was already mentioning before, if you label your protein of interest with a single dye, you will see fluorescence for a certain time tau, and then it will drop to background levels as the fluorophore bleaches. But if we now use this DNA paint approach, what happens is that we get a constant exchange of these fluorophores on the DNA. So even when they bleach, eventually, like, they will be replaced by an unbleached imaging strand from solution. And so you will see some fluctuations to the fluorescence as these imagers bind and unbind and also bleach and end up being replaced from solution. But if you tune the kinetics and the concentrations of these DNA imager strands right, then it will not drop below background level for very long times. And in this case, you see a window of 5 minutes. And yeah, well, does that really work in practice? So if we, for this, we first had a proof of concept data set where we actually use DNA origami that is fixed to a slide, to a glass slide. So these molecules are not mobile, to which we then attach this DNA strand. And in this case, we only had a single binding site. So this is and the long DNA strand. So essentially, we don't have any exchange. This is like really the equivalent of having a single dye on the molecule of interest. So what we classically want to do, we get these very characteristic traces with a single step bleaching of the fluorophore. And after 2 minutes, there's barely any more fluorescent molecules left. Whereas if we have the dynamically exchanging label, even after 30 minutes, we still roughly detect similar amount of signal from this DNA origami than before. And the longest trajectories are also on the order of 30 minutes. Note that in this case, because all the molecules are fixed, we are not limited by any tracking algorithms because the position always remains the same. And this is actually the reason that for these fixed samples, we are really able to get these hour-long trajectories, which we wouldn't see if it was a mobile sample. But so this method works actually, at least from a photophysical perspective, on a chemical kinetic perspective, really very well, as we go from less than 2 minutes to 30 minutes trajectories. So then, of course, we wanted to quantify how much better DNA paint tracking really is. And then we ran into another, not exactly problem, but let's say an issue for comparing fairly across those two conditions. And that is that if we have these dynamically exchanging labels, we can actually get a signal from one molecule more than one time. So if we have a single dye label, once the dye is bleached, this molecule will stay dark forever. But in DNA paint labeling, even if all images dissociate for some time and we lose this molecule, they can bind again at a later point in time, and then this molecule can reappear and be visible again. So for a single tracking handle, we can get multiple trajectories of certain times tau. And that is actually also the reason why, if you look at this plot where we just plot the number of trajectories we obtain from a sample over time, we see that there's a very slight decrease because of phototoxicity effects, or rather photochemical effects to the DNA. But we still are at around 80% of the initial trajectories, whereas for a single dye, this really vanishes to zero over some minutes. So yeah, how to compare fairly across both conditions, taking into account that both these individual trajectory lengths can be longer, but also there are just a lot more of them for the DNA paint tracking. So what we came up with is actually to quantify this in plots that are a bit complicated to read, but they will keep popping up throughout my talk. Where we plotted distributions of the trajectories obtained per particle that last for a given time tau that is greater than the time t over t. So like for every point in time, we ask how many trajectories do we find that have lasted for that amount of time. So the longer the trajectories. So the longer we see the longer this curve is on the x-axis, the longer the trajectories, and the higher the y-intercept, the more trajectories we get of this time interval. So for single dye tracking, this of course has an y-axis intercept at 1, because we only ever get one trajectory per dye. And the half-life in this case, in this plot, is about 1 second. And here you can already see that for this particular, this is just plotting the single trajectories highlighted here in color. So we see that we had one trajectory that only lasts really briefly, and then another one a bit longer, another one a bit longer, and so on, until all the trajectories of this particular handle are essentially plotted, and we go down from 4 trajectories that we had in the beginning to 0 in the end. Anyway, this is a cumulative plot, and on average we get more than 10 trajectories per single DNA origami binding site, and they last for hundreds of seconds rather than seconds. So just plotting the half-life of these trajectories over the irradiance, you can see that no matter how strongly we irradiate a sample, we still get a very good improvement of the overall trajectory length. the higher we go, of course, the better we can actually detect our signals over an existing background, for example. This is why it is interesting to try this at different irradiances, but even at very high irradiances for high backgrounds, we get an average of 3 minutes per trajectory as compared to less than a second for the single dye labeling case. Now, as I already said in the beginning, my group is not really interested just in the fusion of molecules, but mostly in interactions of molecules. And therefore, we were very happy to see that this method works so well, but we also didn't want to stop at just following single proteins. We really wanted to extend this to multiple colors, to be able to do multicolor single particle tracking. And also we wanted to make this method work on live cells, which actually proved to be more difficult than initially expected. But first, about the dual color approach. So in principle, this is rather simple with DNA, of course, because we can just design an orthogonal sequence for a second color to which then different imager strands bind. In practice, this was again also a bit of a fine-tuning of the kinetics to make this work equally well in both colors, but in the end, this is the designs we came up with. We also shifted to slightly longer DNA strands to be able to do this all at 37 degrees. And we can show very nicely that these two sequences are completely orthogonal to each other, so only the matching imager strand bind to the sequence. So we're really sure that whatever we detect in one color is really just the species that we are interested in. And this is what the final data looks like. So we can really see very long interactions in our DNA-PAINT single particle tracking data. This was a plot that I already showed briefly earlier, but I really want to draw your attention to the fact that these particles co-diffuse for more than half a minute. And typically with single dye tracking, which I've done before, which we've always done before, you are happy when you go up to 5 seconds. So this is really a big improvement. And in fact, this meant that we had to also change our analysis software a bit, because we are now filtering the interactions on the localization level before we even track. So that really helps us in stitching together these trajectories of the colocalized particles, because we essentially can already on the localization level decide if there's likely to be an interaction or not. And that really makes the tracking later in a much sparser environment much easier, because we can already throw away all the localizations that are unlikely to be part of the colocalized trajectories. One question that always comes up actually is whether, so these, sorry, I forgot to mention that this is now data of molecules in reconstituted bilayer. So we are looking at two proteins that are brought together by a dimerizer. The protein is called FKBP, and it's just a small molecule that can make these two proteins come together and be a very stable dimer. So yeah, proteins and single proteins in reconstituted supported lipid membranes. And yeah, what we get asked a lot is if this DNA strand that we attach, because it's actually reasonably long, it's certainly much bigger than many proteins you would be interested in. So whether that does affect diffusion. And then, of course, as far as membrane diffusion is concerned, that is mostly governed by the part of the protein that is in the membrane, not so much by the part of the protein that is sticking out of the membrane. But of course, there could still be interactions of this DNA with the other proteins or the membrane and so on. But what we find is that it actually doesn't affect diffusion very much, or at least it affects it not differently than single dye labeling, because what actually was a bit surprising to us is that the biggest differences that we found were between fluorophores, rather than whether we attach the fluorophore to the protein directly or whether we use this DNA-PAINT approach with the long DNA strand. So that is really a bit of a take-home message, I think, also if you, because we all know that dyes influence protein behavior, but when, at least for these diffusion measurements, the it was really quite striking to see how big the influence of the particular fluorophore can be on the motion of the molecule that we track. And this was true for SNAP-tag labeling, where the fluorophore is actually somewhat at least shielded in the protein. So it's not even like just the dye sticking out on the surface of the molecule. So with this, with these nice trajectories, and we were wondering whether we could be a bit more quantitative and really measure 2D KDs in these membranes. And for that, I want to tell you a bit more about the specific system that we studied before I show you the results. Because in fact, we had a bit of an unusual case in the sense that we were looking at a homodimerization reaction. So basically we have these FKBP proteins and they can bind a dimerizer like a small molecule truck. And then that can recruit another FKBP monomer to form a dimer. But if you keep adding the dimerizer, then at some point, of course, you will saturate all the binding sites individually. So you will end up with monomers again. So the interesting thing about homodimerization is that you don't get a classic binding curve where binding just increases. But if you add too much of your homodimerizer, then the binding or the levels of dimercene will decrease to the point that it will go down all the way to 0 again if you have equimolar amounts of them. And this is also true when you use an antibody for dimerization. For example, if you add saturating amounts, then again, you end up with only one antibody being bound to one molecule in a one-to-one fashion. And so we use both this small molecule drug to dimerize the FKBP proteins and also an antibody because we were interested in comparing. The dimerizer has a really strong, very, very high affinity. The dimers essentially are almost covalent afterwards, so they don't come apart very easily. This is different for the antibody, and we just wanted to see if we can pick up the differences in these 2D kinetics of these two molecules. And this is the data we get. So we can really titrate very nicely these binding curves. You can see if we add very little of the dimerizer in the picomolar range, then we start seeing dimerization. And once we go to nanomolar, as I said, this affinity is really very high. Then we lose dimerization again as expected, and we can fit an expression of the binding equilibrium to this data to obtain the 2D KDs. This was actually not previously derived, so we collaborated with someone for this paper. And as you can see, that we find that 2D KDs they have units of densities because it's not concentrations we are looking at, but densities in membranes of course. They are really like 2 orders of magnitude different for the antibody in the dimerizer system, as we kind of expected in advance from their solution kinetics. Now, of course, many or maybe most people are interested in doing single particle tracking in live cells rather than in model membranes. And so the question was really how can we make this DNA-PAINT single particle tracking also work in cell membranes. And the main challenge here is that we like to use TIRF microscopy, so total internal reflection microscopy, to reject the background from the free imager. But at the same time, that means then that we also have to image the cell membrane that is in very close contact with the cover slip. And at the same time, there has to still be space for the images to diffuse into this cleft between the cell and the cover slip, not bind unspecifically to the support, but to exchange with the DNA strands on the molecules in the cell. And to satisfy all these criteria, we really had to screen a lot of different conditions. And of course, you have to find a way to attach the DNA to the molecules in your cell. And we have actually used 2 different approaches. One is based on a GFP nanobody with really high affinity. And so the nanobody is DNA labeled and binds the GFP that's expressed on the extracellular side of the protein in the membrane. And we have also used SNAP-tag labeling, where the benzylguanine that can bind the SNAP-tag is actually bound to the DNA tracking handle. And the only chemistry that we found would work reliably to reject both the background, facilitate cell adhesion, and also imager exchange is as a layer of PEG with an integrin ligand, small peptide, RGD peptide. And this was also, non-covalent coating was not sufficient for this. We actually use a supported lipid bilayer that has lipids that are modified with this PEG RGD polymer. Yeah, and then after also optimizing the fluorophores, this is the kind of data we got. So you now see receptors diffusing on a cell. In this case, that's actually a T cell. And what you can also appreciate is that the background from the free imager dye is actually larger outside of the cell than underneath the cell, because we do get some bleaching or maybe also some restricted diffusion. But in fact, this also increases our signal to noise. So in the end, it doesn't work worse for it. So if we look at the number of trajectories that we see after 2 minutes on a cell labeled with DNA-PAINT, we find that there's barely any difference, whereas for single dye labeling, you can appreciate that many of these molecules have been bleached, and we start seeing the recovery of the molecules from the circumference of the cell, which gives you these characteristic ring-like patterns of trajectories. This is also clear when you analyze more than one cell. So this is like over 3 different samples and about 30 cells or so. You can really see that after 6 minutes, we still have a similar track density for DNA-PAINT, whereas great diminished density, 10% of the start for single dye labeling. And we also improved the average trajectory length compared to single dye labeling by about 2-fold. This is much more modest than the increase that we could find in the in vitro systems. But we do believe that this is mostly to do, in fact, with the tracking and the fact that we are limited to a much smaller area, and that molecules we are also losing trajectories because they diffuse out of our field of view. So that's something that we are also currently looking into to really push our tracking algorithms, because now we also really have the data to make that worthwhile doing. And the dual color approach with the 2 different DNA strands also works really well on live cells. We get, as you can see here, we have one cell where we have this nanobody labeling approach with one color, and in the other cell we have SNAP-tag receptors labeled in another color, and we can really see that labeling is highly orthogonal between these 2 strands. And similarly to the data I was presenting on the in vitro diffusion, diffusion is not really different for the DNA-PAINT approach compared to single dye labeling. So the take-home message really is that I really think that using DNA-PAINT-SPT for interaction studies will improve your statistics. It is actually not harder to implement than single dye labeling, at least. Or even if you use fluorescent proteins such as GFP, it's really straightforward to just add a DNA tagged nanobody to your sample and use that for tracking instead, rather than the GFP signal. As long as, of course, we are looking at extracellular proteins. So this is the big limitation, that because of this DNA exchange, we do have to work, and because we work with live cells, we do have to restrict this method to anything in a cell membrane, on a cell membrane, rather than inside the cell. And yes, as I already mentioned, the influence on diffusion by this label is similar to that of a fluorophore labeling in general, which for any other single particle tracking approach you also won't get around. So with that, I would like to change gears a bit and tell you a bit more about what we want to use this method for in the future. And this is also a project led by Christian, and in collaboration with Hardo Fernandez in Oxford, actually, and Patrick Selle at the NKI, the Dutch Cancer Research Institute here in Amsterdam. And here we really want to understand how interleukin 2 and 15, 2 molecules, signaling molecules in the immune system, are discriminated by the receptor that they both in fact share. So interleukins are generally really crucial for regulating immune responses and for making sure that they are balanced. They are soluble molecules secreted by activated immune cells to essentially spread the information of the activation through the body and to other immune cells. And what's interesting about them is that a lot of these receptor subunits are shared for different interleukins. And in particular, for interleukin 2 and 15, the receptor subunits that carry all the signaling molecules and essentially propagate the downstream signaling, they are really identical. They do have a subunit that is specific to them, the alpha subunit that's specific to interleukin 2 or interleukin 15. But the beta and gamma subunit, they are really identical between interleukin 2 and 15 receptors. And as these receptors assemble, what happens is that constitutively associated JAK kinases will phosphorylate the beta subunit, and then other downstream adapters such as STAT5 can bind and downstream signaling is induced. And even though they share essentially the same receptor, because also gamma beta alone is signaling competent, you don't necessarily need the alpha subunit. This is just making it a higher affinity complex. They have a lot of shared functionality, but they also induce different responses in certain situations. For example, only interleukin 2 will introduce activation, induce cell death in B cells. Interleukin 15 will never do that. And the reverse for memory T cell persistence. So the question really is, how can the same receptor produce different signals using the same pair of signaling subunits in response to these two different ligands? And of course, you could think, okay, maybe interleukin 2 or interleukin 15, they just introduce a different conformation. So, and because the conformation is different, then the downstream events are also different. But it was already found like years ago that the structure of this receptor is really quasi identical on the atomic level, whether interleukin 2 or interleukin 15 is bound. So the green and the green is interleukin 2 and the purple is interleukin 15, and you can see that there's very little difference between those two structures, and also here you can barely see that there are two structures overlaid for the gamma and beta subunit. So being biophysicists, we thought, okay, maybe there's another reason than conformational change. Maybe it is that the receptor assembly differs for both ligands, because there can be slight differences in the affinities, of course, and this could really lead to kind of a discrimination in time, because the assembly kinetics vary between interleukin 2 and 15. Or also, what's also interesting to note is that at least in the vast majority of cases in the body, interleukin 15 is actually presented in cis and not in trans, so not in trans and not in cis, so meaning that there's actually a cell-cell contact. In this context, interleukin 15 will bind to beta and gamma subunit, and this is different for interleukin 2. So essentially, the spatial requirements or the reaction geometry could also make interleukin 2 and 15 look different to the cell. And in fact, because we also have a group at AMOLF that has done simulations of reaction kinetics for a very long time using green function. We looked into this at first at silico. So we just asked what are the kind of species that we would expect for the given rates that have been measured in solution, at least for these receptors. So using the solution kinetics as an proxy for the kinetics in the membranes. And if you just do an estimate based on the solution kinetics, then you would expect that a lot of, so you have all these subunits in the membrane, the alpha, beta, and the gamma, and then interleukin 2 can bind to either just alpha that has a strong affinity for that, or it can bind to all three, or it can bind to beta and gamma alone. So these are the most prevalent kind of combinations that you would expect. And from the 3D solution kinetics, the expectation was that most of the molecules are either in the trimeric bound state, so you have the full receptor, or you just have interleukin 2 bound to the alpha chain alone. But when we actually did the modeling in a 2D system, we found that the species would shift quite a lot. We actually saw that there was a much higher prevalence of the beta and the gamma, so-called medium affinity receptor alone. And actually the species where interleukin-2 was just bound to alpha was quasi non-existent, because essentially all of this was immediately reacting with alpha-beta complexes to form the full receptor. So this was not very long, so any kind of association of interleukin-2 with alpha alone was not long-lived at all. And this is interesting because it could potentially explain why blocking this complex had limited success in the clinic. So it was really focused on the idea that this would be like kind of the first species that would exist on when these molecules interact, when interleukin-2 binds to immune cells. And so now we were really interested, of course, in doing experiments to test whether these are really the species that we find. And as you can imagine, given the presentation that I have already given, we wanted to do this with single particle approach. So reconstituting these receptors in model membranes and studying the association kinetics with single particle, tracking 2-color colocalization, or rather or up to 3-color colocalization, in fact. And for this, my student, it was really the first project in the group, also had to build a TURF microscope. And given that this is a seminar series focused on microscopy and imaging, I do want to share this with you as well, because I think it's we really made a big effort to make this an open source setup. So it is built from components that are quite, in a way, easy to obtain. Some of them were even made by the in-house workshop to make this a very highly functional microscope at a very sharp price. And we have really documented every step of the process, where we get the parts from, how we assemble it, and so on. And we are currently preparing this for submission for an open access hardware journal. But we also, this microscope also has its own website where you can find back all the design files, part list, and so on, and even some nice cut models as to how to put it all together. So if you ever need to build a microscope, perhaps like a TURF microscope, I should say, then this could be maybe of use to you. It also has a focus log and a home-built focus log and a FREP functionality. So when we after years succeeded in making these interleukin proteins, we and label them for single molecule microscopy, we first actually looked at dimerization of beta and gamma subunit in response to interleukin-15 in this case. And while we were really happy that we could actually see clear dimerization, also in a dose-dependent fashion, we were actually surprised that we saw a lot fewer dimers than we expected. So this is at really high concentrations, like upper, almost micromolar concentrations, when the prediction would have been that this all should happen at 10 to 100 times lower concentrations, in fact. So we were really worried about the protein quality. We thought maybe the majority of the proteins is just not functional. So we went back to solution measurements of protein folding, and what you should expect to see in these plots is they are done by like an instrument called nanotemper, where you measure essentially the transition, internal transitions in the protein dependent on the temperature. And if the protein is folded at a certain temperature, it will just unfold. And that will be specific to the protein you study, but at least you should, for a well-folded protein, always see this kind of S-shaped curves where you see a clear transition point. So you can already appreciate there are two lines here that are essentially flat. And interestingly, these were not the lines of the proteins that we made, but the proteins we bought. So like the interleukin-2 and interleukin-15 ligands we bought from commercial suppliers, but we found that the protein was of really poor quality despite all the promises to the contrary by the manufacturers. And when we did some kinetic KD measurements with the same proteins, we also found that the KD was about 1,000-fold higher, or the apparent KD was 1,000-fold higher than the literature value, really suggesting that most of the protein is not functional. And this was just in a sense, a bit of a negative result, but just to share, to be aware that what you buy or what you think you buy is not always what you get. And it can be just good to keep this in mind. And with that, I really want to thank my group for fantastic 4 years, in particular, Christian, for doing all the work on the projects that I mentioned. And our collaborators for all the help on the DNA-PAINT, Petra Schwille and Ralf Jungmann and Johannes Stein, who was the person involved on the lab side. And for the Interleukin work, Ricardo Fernandez, Patrick Selly, and our colleague Peter Reims involved at Armolf. And if you want to build a TURF microscope or move to doing DNA-PAINT-SPT measurements, then please go to our group GitHub page for more information or get in touch for advice. And with that, it only leaves me to thank you for your attention, and I'm very happy to take any questions you may have. Thank you.
Thank you very much for the very clear and super interesting presentation. I'm glad that I came back for this one, as I've been a bit absent. So before the question and answers, we have our magnificent Mentimeter quiz that Stephanie prepared.
Yes, that's very true. So I should share my screen. So this gives the speaker a short moment of relaxation, but of course, I think, sorry, you need to actually unshare your screen.
Yes.
Thank you. So I will share my screen, and then, okay, that's always the moment which I find really unpleasant because, okay, I need to share one screen, and then I should be, that's it. Way. Okay, so is that okay?
Yeah, it's working.
You see my magical quiz?
Absolutely.
Very poignantly, I was supposed to be in Berlin, but yesterday I got stuck at the airport because I was in a snowstorm on the runway at Stansted when it got closed down. And so everybody, I think here in the UK at least, is a bit freezing. So I just ask you, how hot or cold are you to begin with? And you can tell me. It's also poignant in that people who know me know that I'm changing my job. And I have actually started also, well, I also shifted what I do because I joined a climate tech startup from Cambridge who's moving to Berlin called Carbon 13. So these are the two connections to this slide. So I think I will be a little bit of a visitor from time to time, but I hope, yeah, this will be continuing and I can. So can you, I don't know, does it work? This kind of Mentimeter thing never quite works because I should be able to see results.
I think you see the average.
Oh, yeah. No, it always throws me, this Mentimeter.
That is why we like when you do it.
I know, it always makes me very nervous. I think, oh, okay. Well, see, there you go. So I think we saw the average. I don't know what, I'm sorry, I can't. Please join. Come on, this is respect to the Imaging One World community. Please join. Come on, more people. Okay. A few more people joining, please. Okay. Okay, we start now. But it will be quite nice if you listen. So these are provided by Kristina. Why can classic DNA paint not be used for single molecule tracking? And you have 3 answers. And I'll let you choose one of them and see if you get it right. Okay, quite a few people got it right. Moving on to the leaderboard. There's always a thing between the leaderboard and the actual result. Okay, so we have like Storm Mick, who I think this is an insider very much, kind of leading the crowd here on this one. So thank you very much. Let's do the next one. And next slide, the next question. If you have a comment, I mean, Kristina, please feel free if you want to comment. But I think it's, you just go through it. Question. Okay. Finding the optimal DNA sequence needs consideration of, and then binding kinetics, bleaching rates of fluorophores, or both of the above. 7, 5, 3, 2, 1 second. Time's up. So that was actually a difficult question. People should have known from your talk, but maybe they didn't listen. So both of the above, but it's also, I guess, a bit of a trick one. So both of the above includes option A, binding kinetics. So the right answer.
I wasn't aware of the display when I
oh yeah, it's kind of all kind of quite adventurous. Okay, this is the leaderboard. Now see, I think Leonor and Mars have overtaken Storm Mick, and Montana is actually currently, because you also win points or gain respect of the Mentimeter if you're fast. So I never know, I have to press enter or next, and I always get it wrong here, but question 3 of 5, press enter, so answer fast to get more points. The relative improvement gained by DNA-PAINT-SPT over single dye SPT in the best case scenario is around 30 times, 100 times, or factor of 3. Okay. And the time is up. Okay, so around 30 times, not around 100 times. I was very optimistic, but I think that around 30 times is pretty good, you know. So the next leaderboard race. Nick always gets very excited about the leaderboard, so I left it in just for Nick. He's always a bit like Leonor, still the fastest. What happened to Storm, Nick? Come on, get going, get going. Question 4. And again, enter, answer to get fast, to get more points. Using DNA-PAINT-SPT on live cells requires a snap tag on protein of interest, careful surface chemistry to reduce unspecific binding, or a red fluorophore. Okay, I'm curious. Everybody, careful surface chemistry to reduce unspecific binding. Excellent. Again, the magic of the leaderboard, showing how people race along, and still Leonor is kind of seems to, but very shortly, very closely followed by Physios. I think some people have very imaginative names here. Some are kind of made up by meeting Mentimeter automatically, but I think that was a made up one. So answer first to get more points on this question. Does labeling of for DNA-PAINT-SPT influence diffusion? And the answer were always, never, only for some fluorophores. That's your last chance to win this race. The last question. 3, 2, 1. Time's up. And it's right, only for some fluorophores. It's always the safe option if you say, Now see the leaderboard race, who's actually now winning. Magical Rhino, Alex Stormick. I like Magical Rhino.
Yeah.
Oh, but Leonor has actually won. So Leonor, you won the race, and you're going to be sent per snail mail, probably a proper fold scope microscope. Initially, when we started this, it was obviously telling people about microscopy, giving them opportunity to do microscopy outside the lab, because we started it in lockdown and nobody had access to microscopes. But if you have children, if you like to kind of
They're still
share
fun to play with, I have to say.
share the kind of knowledge, I think it would be fun. Should I unshare? Is it unshared? Did I
Yes,
unshare?
yes. It's
Yeah,
all
it's
good.
already okay.
So today actually will be a special foldoscope, and that can do single particle tracking. Okay.
Yay!
Can I have one too?
Actually, I thought you were providing it as an open
That's a
source.
disappointment.
We may
I was
send you one
particularly interested
too.
in the lasers.
All right,
So,
next time we'll work on that. Not just
Um,
open science for like building your own fancy, still more than 100K microscope, but actually a foldscope with the same properties. That's the new challenge for the next PhD student.
That's a bit tough.
It will be a very long PhD. So is there any question from the audience? I mean, you can unmute yourself as well if you want to.
Yeah, people want to kind of just ask the questions. I mean, there was so much information. It was really lovely.
Hello.
Oh,
okay, yeah, please.
Great talk. I wonder if it's known, or if you can go into a little bit of more details, why the single dye labeling affects diffusion so much. You showed the graph with ATO488 and AF647 and JF549A, and those were like all over the place.
Yeah, we were actually also surprised to see that. I mean, it's not that we were not expecting there to be a difference per se, but that it would be that huge. It really came of a bit of a surprise to us. So one thing that I should say is that, of course, if you track anything in a membrane, then you also have this kind of difference of like there's also a hydrophobic, say, environment. And I think that it is clear also from other people's data that certain dyes are just much stickier or much more hydrophobic than others, and they tend to interact more with the bilayer as well or the lipids, and then you can get an effect on diffusion from that. But yeah, I mean, no, we don't really have a very good explanation nor a good way to predict which dye will behave in what way. But certainly it's something to watch out for.
Great, thank you.
okay, while we wait to see if there is anyone else that want to make a question, I have a question. It's rather general, because on the specific you have been so clear that whenever I had a question, you already answered. Next slide. So, but I do have a general question. It's about every time that we try to do things that are a bit more that are open source, and clearly you're doing a very good effort in that direction. There is always a part that, you know, it is simple, but it is not as simple as we wish. I wonder if you could tell us which would be the most difficult part to either maintain, you know, something like alignment and so on, or to build in your setup.
Yeah, I think that what really is not so easy is the fact that we did some custom electronics for this. So some of the hardware is actually connected, I mean, to the computer, of course, like for everyone, but we are like kind of an extra piece of hardware that switches faster and also coordinates between different boxes. And we have, of course, shared all the instructions on how to make this, but I think your average PhD student will probably not be able to do this. And so in a sense, to build the setup, at least the way we did it, and also for certain other parts of the microscope, there was some custom-made electronics involved. You'd need a workshop available to you to do this, or you would need to outsource this, which is probably also an option. But then it starts to become probably less interesting. So there's a certain amount of technical expertise for that required. I think for everything else, it's I think it should be really doable for someone with a little bit of optics experience, or someone who has someone to ask for certain questions around in the lab, to really build this microscope, and it would work just as you would expect. In terms of keeping the alignment and all of that, because we really minimize mobile components, it actually is really stable. I mean, not that you never have to align it, but it's really pretty stable.
Yeah, no. I mean, I guess that perhaps, you know, is not a day-to-day thing to have to do this. Yeah,
no,
good.
yeah, indeed. Yeah, definitely not day to day. No.
And overall,
I mean, they're quite. Sorry.
no, go, Stephanie. Stephanie, go.
I mean, there are a few efforts to go away from like the classical microscope setting at all. No, I mean, for stability reasons, or also commercial systems.
That's what we do. I glossed over this, but we actually don't use a microscope body for that reason. For example, it saves money at the same time, because they are, of course, one of the bigger pieces of expenses. Like, but also then you get a piece of equipment that you want to have serviced by an engineer from that company rather than yourself. And also you get drift and there's functionalities you don't need. For example, an objective revolver for turf, you don't really need most of the time, at least most people don't need it. They want to use this one single objective all the time. So that has then mechanical components that can introduce drift that are completely unnecessary and so on. So we actually moved a sample. And for that we have like a piezo stage, which is also really very stable. So that's one of the things that we did to make it more friendly, also to maintain in terms of alignment. In fact, I mean,
like the I mean, ONE have like a very small box, kind of for super resolution imaging, which I think is a very clever way to get away from the kind of issues of drift. And
I've never tested one of their setups, but I'm very curious about them.
Yeah, they're very good actually. I mean, I think you probably have obviously many other things attached, and channels and flexibility and kind of freedom in your setup, but
that's always a trade-off, of course. I mean, because that's indeed true that we included a few things that we just like, for example, this frap or something like this that we really need for the bilayer. Some other people may not like it, but the other thing is also it's really modular, so you could really like also decide, I don't need this component, and it would work if you just take it out. So there's a few of these things that are really like a module you can have or not have, but it would not influence anything else.
And I guess one question which people I'm sure will come across, or you come across straight away with biologists, they say, but how deep can you actually image into systems? And that's always then the challenge. Usually you give them fast and live and high resolution, and then they say, but my sample is very thick. Maybe you can comment on that a bit as well.
Yeah, so I think this is absolutely. No, so I think that that is always a challenge because of background rejection, and we haven't really tried pushing this very much, but I would actually be surprised if at least the way we implement it right now with the concentrations we have to use right now, I don't think we could make it work for thick samples. So we would be restricted to this classic 100 to 200 nanometer range close to the cover slip for now. I'm not saying it wouldn't be possible, but it would certainly require a lot of adjustments again to the method, because we for DNA-PAINT we actually use a reasonably high level of imager strengths, which gives us also quite a high background, and that means that we really have to use turf. We can't use, for example, confocal microscopy. I mean, like, I know that some paint people like Ralph do that, and they can do it with spinning disc confocal also for certain samples, but the conditions they use would not be compatible at the moment, at least with what we do.
Yeah, thank you. So, Alex was writing fluorogenic DNA paint into the comments. Do you want to ask the question properly, or you?
Oh yeah, the fluorogenic one. Yeah, that is something we have also been thinking about. Yes, so basically he means that when we have, I think he means, he can correct me if I'm wrong, I think he means that essentially the imager probes only turn fluorescence once they bind to the DNA, and therefore you have a lot less background. Yes, certainly something to look into, because I can only see it would benefit this method. Yeah. I mean, maybe the simple answer is for certain things that you may want to do, this is not even necessary. You can do without, but I'm sure you can be better with. Yeah.
Okay, excellent.
Here's the cold scope. You can.
We don't see.
Oh, sorry, it's not.
Yeah. So I think it's time to go to an end, because it's already 2. Well, I would like to thank Stephanie for, she's a friend, and I wish you well for your new adventures, and hope to see you not only around personally, but also around on Imaging One World. Okay,
we'll pop in from time to time.
It was a great adventure, this one as well.
Yes.
And thank you everyone for having joined. Thank you for who is watching on YouTube, and of course, thank you, Kristina. It was great to get to know you today.